noticing you. My name is SB. Whenever I do a dance, I feel tired. Then after, I feel okay. It's like something is replenishing my energy. Let's find out.
After all of that research, I know why I get my energy back after spending it. This is because of cellular respiration. So what is cellular respiration? It is a catabolic pathway, which means it breaks down complex molecules to yield energy. The complex molecules to be broken down would be glucose from the different things we eat, such as cake, cookies, and carrots, and more. Cellular respiration happens in the cell, with specifically the cytoplasm in the mitochondria. There are two types of respiration, aerobic and anaerobic. This one needs oxygen while this one doesn't. Harvesting energy from glucose is in three metabolic stages: glycolysis, pyruvate oxidation and the Krebs cycle, and electron transport chain and chemoosmosis. Before we go to the different stages, I would like to introduce to you Mr. ATP. Mr. ATP, or his full name, adenosine triphosphate, has the capability of transforming energy for cellular work from its phosphate bond. Now, Mr. ATP will be produced by the following stages in order to transform it to yield energy for cellular work. First up, glycolysis. So, glycolysis is done in the cytoplasm and has two phases: energy investment phase and an energy payoff phase. In energy investment, the glucose is subjected to enzymes that transfer two phosphate groups from A to ATP, resulting to its cleaving into two different 3-carbon sugars called G3P or glyceraldehyde 3-phosphate. In energy payoff, the G3Ps are oxidized by NAD, forming NADH, and another phosphate group attaches. Now this dephosphorylates, letting go of the phosphate group, and transfers it into an ADP to become ATP. After, water is extracted from the molecule, then dephosphorylates again, transferring another phosphate group into an ADP to turn into an ATP, and turns the molecule into pyruvate. In the end, glycolysis yielded negative 2 ATP in the energy investment phase, and 4 ATP, 2 NADH, and 2 pyruvate in the energy payoff phase. All in all, glycolysis gave 2 pyruvate, 2 NADH, and 2 ATP. Now let's go to the next stage. Now, this pyruvate oxidation is like a transition of locations from cytoplasm to the mitochondria. First, the pyruvate releases its carbon since it is oxidized, making it carbon dioxide. And because it is oxidized, it releases electrons, making NAD to NADH. Then coenzyme attaches to the 2-carbon, producing acetyl-CoA, with which the coenzyme helps in transferring to the citric acid cycle. This stage yielded 2 NADH and 2 acetyl-CoA. Whee! Now this is the citric acid cycle or the Krebs cycle. The cycle starts with acetyl-CoA becoming a 2-carbon acetyl group with the leaving of coenzyme A. The 2-carbon acetyl group will be added to a 4-carbon oxaloacetate, and it becomes a 6-carbon citrate. With the help of enzymes, citrate becomes isocitrate, which loses a water molecule. Then isocitrate will be oxidized, which reduces NAD to NADH and loses a carbon, becoming carbon dioxide. It is now called the alpha ketoglutarate. Alpha ketoglutarate then loses another carbon because of oxidation, becoming carbon dioxide, and also reduces NAD to NADH here. Now the coenzyme A comes back, attaches to the succinate, and becomes succinyl-CoA. Succinyl-CoA is displaced by a phosphate group which transfers to a GDP to become a GTP, which generates also ATP. Coenzyme A leaves succinyl-CoA and becomes succinate. From succinate, it is oxidized with 2 hydrogen, transferring to the FAD to become FADH2, turns the carbon molecule into fumarate. Water is added to fumarate, rearranging its bonds, turning into malate. Malate is then oxidized, reducing NAD to NADH, turning it into oxaloacetate. Then the cycle begins anew. The cycle runs 2 times per glucose molecule, and it yields 2 ATP, 6 NADH, 2 FADH2, and carbon dioxide. Now let us go to the final stage. The produced NADH and FADH2 are actually used for the synthesizing of ATP, especially their electrons. This part is done in the inner membrane of the mitochondrion, wherein the cristae increases the surface area for electron transport chain. For NADH, it starts in complex 1 of the chain where it does a redox reaction to transfer electrons. This goes on to less electronegative electron carriers. For FADH2, it started in the lower electronegative complex 2, although it gives the equivalent number of electrons within ADH. The last electron carrier is oxygen, which forms water, with the electrons and pick up hydrogen ions. For chemiosmosis, the ATP synthase is powered by the energy of the electron transfer chain, specifically the difference of concentration of hydrogen ions on the opposite side of the mitochondrial membrane. In the ATP synthase, the stator lets the hydrogen ion in and bind to the rotor, which it spins. After a spin, it is released in the mitochondrial matrix, and the spin causes the internal rod to spin, which activates sites for the producing of ADP into ATP. This entire process has yielded 26 or 28 ATP, which totals up to 30 to 32 ATP yielded by one glucose molecule. So in order to produce energy, you need your food and your oxygen. But what if there is no oxygen? Without oxygen, the cells will undergo anaerobic respiration and starts at the end of glycolysis in the cytoplasm. The way is called fermentation, which is an extension of glycolysis that allows continuous generation of ATP. First is your ethanol fermentation, wherein carbon dioxide molecule will be released from the pyruvate, which converts it into acetaldehyde, and it is reduced by NADH yielding ethanol. The other one is lactic acid fermentation, wherein pyruvate is reduced by NADH, regenerating NAD, and it produces lactate or lactic acid. This is commonly produced through strenuous exercise, where the muscle results in lactic acid fermentation because of the shortness of oxygen. And what's that?
Oh, talk about the woozy. That was fun, isn't it? We know now how the food that we eat is being transformed into energy. Now, once again, my name is SP, and that's a wrap.